Welcome back to Enzyme Regulation, Control of Enzyme Activity. This time, we will discuss how regulation can occur at the level of DNA. To get the most out of the videos, I recommend watching them in order. This is a reminder that this presentation is Dr. Johnson's intellectual property. Ultimately, the longest-term solution to regulating the amount of enzyme activity available is to reduce or increase the concentration of the enzyme. The longest acting method for achieving this is to regulate gene transcription. Genes for a metabolic process are usually organized into an operon. There are two ways the operons can be regulated. They can be induced, which increases the synthesis of mRNA and therefore the enzymes that they encode. This induction usually occurs as a result of the presence of a metabolite. For example, the lac operon is inducible. Other operons are repressed rather than induced, resulting in the decreased production of mRNA and therefore enzyme. Like induction, repression occurs as a result of the presence of a metabolite. An example of a repressible operon is the His operon. The lac operon is comprised of 4 genes as well as the control sites, P for the promoter and O for the operator. A repressor, encoded by lac-I, is normally bound to the O section of the operon, preventing transcription from the promoter region by being a physical barrier so that RNA polymerase cannot open and unzip the DNA double helix. An inducer binds to the repressor, inactivating it so that it no longer binds to the operator. This allows transcription to be initiated. The inducer for the lac operon is normally 1,6-allolactose, but other sugars can also be used, especially in lab settings. The repressor is an allosteric protein with at least 2 conformations. Another way in which the lac operon is regulated is with the catabolite activator protein, or CAP. Glucose prevents transcription of the operons for many other sugar metabolizing proteins. This occurs because cells have a preference for metabolizing glucose via glycolysis. While other sugars can also be metabolized, this requires additional steps and the enzymes to catalyze them so that the metabolites can enter glycolysis. Therefore, if there is plenty of glucose available, there is no need to use these other sugars for energy, which means that the enzymes required for this are also not needed. Glucose mediates this downregulation of the other pathways by decreasing the cyclic AMP levels in the cell. We'll see why this is important in a moment. The flip side of this is that if glucose levels are low, the cyclic AMP levels increase and initiate transcription. So, what is the role of cyclic AMP? When cyclic AMP levels are high enough to be near the Kd value, cyclic AMP binds to the catabolite activator protein, which results in dimerization and activation of the protein. The activated protein then binds to DNA, where it assists RNA polymerase binding. This is how transcription is activated. Ultimately, for a really high level of transcription of the lac operon, both a specific inducer for the repressor protein and cyclic AMP must be present. The lac operon is not the only operon controlled by CAP. It and others controlled by CAP are part of the global regulatory circuit. Here we have a schematic illustrating the 4 different regulatory circuit options. Operons can be under negative control, with a repressor protein controlling transcription, or they can be under positive control, with an inducer protein controlling transcription. The repressor and inducer protein are further activated or deactivated by binding of a small molecule, which changes the conformation of the regulatory protein. If the binding of this effector causes induction of the gene, the effector is a co-inducer. If the co-inducer binds to the repressor of a gene under negative control, the repressor is deactivated and transcription can occur, like we saw with the lac operon. Alternatively, binding of the co-inducer to an inactive inducer protein could activate it and facilitate transcription. This is depicted in the top right rectangle. Catabolite repression is an example of this type of control. If the effector causes repression of transcription, it is a co-repressor. For genes with a repressor, the co-repressor's role is to bind to an inactive repressor to activate it. The activated repressor binds to the operon preventing transcription. The trip operon is an example of this type of regulation. For operons with an inducer, the role of the co-repressor is to bind to the active inducer and render it inactive. The inactive inducer cannot bind to DNA. This has been a brief overview of regulation at the DNA level. Next time we'll look at the RNA level.